Okay, let's get started. Well, good afternoon, everyone. Welcome back to the Kapner Research Council webinar series. As you may recall, we converted the CRC Coffee and Conversation series into a webinar following the COVID-19 pandemic and subsequent campus shutdown last spring. We kicked off the fall webinar series with Dr. Parag Chitnis, Acting Director of NEPA, late last month, and had Dr. Mary Hendrickson as our second speaker 2 weeks ago. Today, we have one of our newest faculty members, Associate Professor Dr. Kihoi Lee from the Division of Animal Sciences, as our speaker. Dr. Sarah Lowe, Chair of the Kapner Research Council, will make the formal introduction of the speaker. But before then, I would like to remind everyone to post if you have any questions in the Q&A box during the talk, not in the chat room. I will be moderating the Q&A session today as Dr. Lowe has to leave for another meeting at around 4 o'clock. So without further ado, Sarah, the floor is yours.
Thank you, Dr. Jose. I'd like to introduce Dr. Kihoi Lee. He graduated from Seoul National University and then received his MS and PhD from Purdue before coming to Kapner and University of Missouri for postdoctoral training 2011 to 2013. While at University of Missouri, Dr. Lee generated the first immune deficient pigs that could support growth and proliferation of human-induced pluripotent stem cells. We're pleased that Dr. Lee has returned to Kapner as an Associate Professor of Animal Sciences after having been an Associate Professor in the Department of Animal and Poultry Sciences at Virginia Tech. At Tech, Dr. Lee's lab focused on early embryonic development using the pig as a model. His group generated various types of genetically engineered pigs for agriculture and medicine. One of his main research interests was to identify the mechanism of epigenetic reprogramming by oocytes after fertilization. His research has been funded by prestigious agencies including USDA and National Institute of Health. Dr. Kihoi Lee, so pleased to introduce you today. So glad to have you here in Kapner, and we look forward to your talk, Use of Livestock Models Under the Genome Editing Era. As a reminder, please put your questions in the Q&A. Dr. Lee, I turn it over to you.
Thank you. I see, I don't know if it's a technical issue. I still see some issues with my internet. Hopefully it will remain stable.
It's been one of those days.
Yeah, exactly. Is it good? Okay, so the title of my presentation today is Use of Livestock Models Under the Genome Editing Era. And I picked this topic because, well, it's my interest, but also we actively work on utilizing these models. And I wanted to provide kind of a big umbrella of my program. So some contents might be too brief, which I'll be happy to address those in questions, or now everybody knows where I work, so feel free to grab me and ask these questions. So for just and Dr. Lowe provided a great introduction. So just to repeat some of my backgrounds. So I received a bachelor's degree in animal biotechnology from Seoul National University. So I'm originally from Seoul. Then after obtaining my bachelor's degree, I wanted to explore the world and be exposed to a bigger world and scientific area. So I joined Purdue University to earn my master's and PhD degree in animal sciences. After obtaining my PhD, and the thing I wanted to do was to work with pig models, and the leading institute, and still is University of Missouri. So I joined Dr. Randy Prather's lab to get my postdoc experience. And I joined Virginia Tech to open my lab and worked there for about 6.5 years. at Missouri. So I have 3 compartments that kind of constitutes my my lab, which is biotechnology, embryology, and reprogramming, and emphasis in epigenetics. And hopefully at the end of my presentation, you can kind of understand, okay, this is what he's doing. And these all 3 compartments kind of work together instead of studying 3 aspects separately. And my main interest, again, is livestock model, because I truly believe these are better models. I think throughout all my career, I worked on a pig or bovine model. Not that I despise rodent models, but I truly believe that these models are better, and hopefully I can continue to work with these models throughout my career. So livestock models, and the first I would like to begin with, hey, why livestock models? Why are they better, or why is for example, and it was actually at University of Missouri, the first cystic fibrosis model has been developed in pigs. And those models greatly or largely recapitulated human symptoms, whereas rodent models did not really capture all the signs of cystic fibrosis. And other than these disease models, I'm interested in modeling pregnancy using these animal models, because sometimes the durations matter. And modeling pregnancy, again, for example, in rodents, the gestation period is only a few weeks, whereas these domestic animals have a much longer gestation period, so that we can watch how the pregnancy goes on, which is really difficult to study in rodent models. And another aspect that I'm more in particularly interested in is to study events of early embryo development. So again, one of the aspect of my program is embryology, and I'm interested in how these embryos start from a single cell and become these series of embryonic stages, and then ultimately become multi-layer of cells and structures that eventually consist an organism. So that's something that I'm really interested in. And besides biomedical application for agriculture purposes, these animals are an important source of protein for meat production. For example, in 2017, 26.3 billion pounds of beef and 25.6 billion pounds of pork were produced and consumed in US. And again, these numbers indicate they're very important animals in agriculture. And as a land-grant university, I think it's important for us to follow this mission and make sure that these animals will be studied and used so that we can secure sustainability of these animal production. Then why not use these livestock models if they're so great? Again, I'm a little bit, sorry, I'm a little bit distracted because I don't know if there is an issue with internet or not. So, because I keep hearing these beeping sounds. So why not using these animal models? So the biggest concern that I have, at least related to my area of research, is low efficiency in genetic modification. Why is that important? Why is it important to modify the genome of animals? Well, so with the development in bioinformatic technology, now we know, and genome in general, now we know certain traits or disease, they're really linked to genetic level. So we now have a much greater understanding of how genetic impacts phenotype, basically. So if we have a question, right, so it doesn't matter in which area, it could be reproduction, it could be nutrition, it could be in any other field. And we want to know what this gene X does. And in traditional way, it's really difficult to produce these, if hypothesis is to study this in people, to generate these models and answer our question. So alternatively, because the models are readily available, people tend to use rodent models and try to translate that results into the animal of interest. So in this case, it would be pig. However, just like anything else, rodents are very different. So the results we get using rodent models often does not translate into what we want in pig. So it could be very different. And in parallel, same scenario can be applied where if we try to answer a question using rodent models in human disease, often cases the results does not represent what's happening in human body. So that is an issue that we face. Development of genome editing technology can solve a lot of these issues. And hopefully I can convince you at the end of this, this presentation that, oh yeah, it is more affordable or more practical to produce these animals and use those for different experiments. So genome editing aspect can be overcome with a recent development. However, large animals still again have some disadvantages compared to standard laboratory animals because they have longer gestation period and much longer compared to rodents. And breeding just takes time. And often cases, if someone is just mainly using rodent models, it's really difficult to convince them to jump into a better model or pig model in this case, because they just can't wait. So if I tell someone that, well, I can make a model that you're interested in, and I will have these models for you in 2 years, they're not going to call me anymore. So I need to find a way to make these models in more timely manner so that they can answer their biological questions. Another, I guess, obstacle, it's not a limitation, but obstacle is infrastructure and husbandry. It is more expensive to maintain large animal models compared to rodents. You can't put pigs in a box or pigs on a shelf that just take out whenever you need them. So you really need a designed planning and how to use these models and time it well to use these models effectively. And I'm glad that I joined MU because we have great infrastructure, and I've been told that will continue, so that will be the leading group in working with these models. So genome editing system, I don't want to go over too much background because a lot of content is now more or less standardized. So just in brief, these genome editing systems, mostly, not all genome editing systems, but they rely on locus-specific DNA break. So once they introduce locus-specific DNA break, then we can utilize that opportunity to introduce site-specific modifications. And these genome editing systems are called as engineered endonucleases, homing endonucleases. People use different names. And the types of genome editing systems has been introduced are zinc finger nucleases, TALENs, and CRISPR-Cas9 system. It really dramatically changed the efficiency to produce genetically engineered livestock model. And just to share personal experience, I could not make any models during my PhD, and in part because I struggled during my PhD, and I think it added a year or two to my program. However, once I start to utilize these models, production of these genetically engineered livestock was much more feasible. And I don't want to go into too much technical details, but we can utilize these systems to introduce site-specific modifications in cells and then generate animals through somatic cell nuclear transfer. In other words, cloning, or we can now directly inject these genome editing systems, in particular CRISPR-Cas9 system, to introduce site-specific modifications. So this is the image where I perform nuclear transfer, cloning, and this is an image of introducing CRISPR-Cas9 system into developing embryo. Technical-wise, direct injection is easier, you can say that. However, both methodologies have advantages and disadvantages, limitations. So it all depends on the end results you would like to have. And end result being that we produce precious pig models that can be used and can be sold at high cost. So first study that I would like to introduce is the design of pig models for biomedical applications. And in particular, I utilize immunodeficient pigs, we call it skid pigs, for cell transplantations. So use of skid pigs for regenerative medicine. So these pigs can support growth of human stem cells. I was here when we first made the report. These pigs, we produced pigs lacking adaptive immune system and introduced human iPS cells, and they resulted in teratoma, which indicated that these pigs can support a growth and differentiation of introduced human stem cells. So that really indicated us that these models can be used to generate humanized models. The examples are if we introduce hematopoietic stem cells, these pigs are in theory can produce human antibodies that can be used in clinics as a remedy. Cancer models, we are currently working with oncologists to introduce human cancer cells and grow them in these pigs. HIV study, these pigs don't have lymphocytes, so that represents acquired immunodeficiency. Stem cell therapy, we can introduce stem cells and see how they grow before that goes into a patient. So as a translational study, to capture any unexpected adverse effect, it is very important to use these livestock models or large animal models, if you will. And one of the topics we are currently focusing on is organogenesis, where we introduce human tissues and let them form into a mini organ. So once I generated pigs lacking adaptive immune system, next goal was to generate a full skid pig, if you will. And by removing 2 genes, I should be able to reach that goal. And we started to generate RAG2 and IL-2RG knockout pigs because they would lack major lymphocytes, B cells, T cells, and NK cells. And these are similar, their phenotype will be similar to NSG mouse, which is a leading. And again, these animals are ideal for cell transplantation to grow human tissues and cells. But especially these pigs are really almost impossible to generate through breeding because RAG2 or IL-2RG alone leads into immunodeficiency that we can't really breed pigs. So these pigs fail to thrive, so we can't really breed them. And the housing, it's almost to a point where we need to keep them in a bubble if we do not want them to be exposed to potential infection. And at time requires to breeding is just a nightmare. And we try to do some calculations, say, okay, if we want to make these pigs through breeding, avoiding immunodeficient phenotypes, so maintain all these founders heterozygous, it would require about 256 pigs to generate 1 pig that we want. So one of the studies we did, we produced 17 piglets. Well, to reach 17 piglets, we would have to produce 4,000 pigs, over 4,000 pigs. I don't think any farm would like to do that. So that was a trigger point where we decided that we cannot make these through breeding. We want to make these in a more straightforward, rapid style. So we decided to introduce CRISPR-Cas9 system into developing embryos. So this was the first report of NSG-like skid pigs. So we targeted both RAG2 and IL-2RG simultaneously and injected CRISPR-Cas9 targeting both genes into developing embryo. And by performing embryo transfers, we generated these NSG-like skid pigs. And their initial phenotype, their typical phenotype is athymic. So you can see that here, the pig lacking RAG2 and immunodeficient IL-2RG also lacks the formation of thymus, and lymph nodes, they're pretty much gone or very underdeveloped. And when we looked at their phenotype by performing a FACS analysis, and you can see their B cells, T cells, and NK cells are dramatically reduced compared to the wild type. You can see the numbers. And my collaborator was interested in following up events in the gut. So we looked at ileum, but same trend was identified in circulating lymphocytes. And we performed additional histology to ensure that these skid pigs do not have lymphocytes. And you can see here, B cells, T cells, and NK cells are all missing in SCID pigs. So after generating these animals, then we decide to use these models as a patient-specific disease, to follow patient-specific disease. So again, these pigs can allow growth of human tissues. So if we introduce patient-specific cells, tissues into these pigs, then these pigs will allow tissues to further grow. And we have a project where we try to generate mini organs from these pigs, or we use these pigs to find cancer treatments. And again, I mean, in brief, even there are many approaches to identify a proper remedy for cancer patients. We just cannot give these patients, let's try 100 things and see which one work. The patient will probably die by them. So, but then if we generate these animals, we can test these remedies in these pigs first before going to the clinic. At least that's the idea. Again, working with these pigs are challenging because they're immunodeficient. Actually, my students really loved working with these pigs, wearing all these gears. I don't know about the pigs. I think pig was screaming. So this is a transplantation of humans. We work with Cincinnati Children's Hospital. So this represents a human gut. And this is where image of us performing transplantation studies. The black arrow indicates where we placed human organoids. And about a month later, we were able to harvest a human organoid that got bigger. And the histology indicates it represents a gut structure. So that success really encouraged us to go further. But then we lost a lot of pigs through performing transplantation. So we actually decided to do everything as sterile as possible, so that these pigs were housed in gnotobiotic housing, which means it's a germ-free environment. And then even transportation to surgical facilities, sterile surgical facility, they were housed in a bubble. And so after doing that, these pigs were fine. And then this is an image of pig received human intestinal organoids. And their growth was fine. They did not become sick and die. And because we labeled our organoids this time, you can see the green organoids glowing in pig body cavity. And you can see that human organoids attached to pig gut, and it glows like, you can see kind of yellowish color because it's tagged with green fluorescence protein. So this is an image of human organoids, got big. Typical size of human organoids we found was between 1 centimeters. The biggest one was about 2.5, which is pretty big. In the beginning, when they entered the pig, they grew quite a big. And the histology indicates that they are positive for human nuclear antigen, so that confirms that this is from human origin. And we struggled because they're so sensitive to these inflammations or infections. But then, as long as we provide a sterile environment, we were able to transplant or engraft these human organoids. So now we know they can grow these tissues. And the next step was to, and we have a grant where we try to engraft human cancer cells. And similar to the previous approach, we produced these pigs and introduced human pancreatic cancer cells. And you can see their weight was still growing, and pig tumor grew over time. And histology suggests that this was an ultrasound image of us looking at the tumor. And histology indicates that this is indeed a pancreatic tumor. And when we looked at histology, it's the interesting aspect is that when we use these models, the outcomes of engraftment or transplantation is different from when we use NSG mouse model. So we were assuming that, well, the outcome will be the same and we will be able to grow these engraft in a bigger manner, but it's kind of beyond that. They do engraft, but we do some differences, which I don't think the differences are bad. Actually, I think these are really indicating us that we need a comparative study. And we can actually use these to study graft versus host disease. So that kind of, again, I briefly touched on the application of pig models that we're currently using. And I want to switch your gear a little bit and provide some information on how these tools can be used to study events in embryo development. And that's a beautiful embryo that we have. And one of my interests is elucidating epigenetic reprogramming. So there is a dynamic change in DNA methylation during pre-implantation embryo development. So embryo forms after fertilization, and it becomes embryo, what we call blastocyst. When we see blastocyst in lab, we're so happy because that, and eventually these blastocysts will become a pig. So by understanding how these events occur, hopefully we can develop a way to produce better pigs, healthier pigs, or more pigs. And another aspect that I'm interested in is because oocytes have a very special ability to reprogram or remodel the genome or epigenome of sperm and oocytes. If we understand the mechanism, that we can use that mechanism to improve our efficiency in somatic cell nuclear transfer, which is cloning. But all these reports are mainly in mouse, not even other rodent species, and no comparative studies have been reported. So again, I won't go over too much in detail. I found not many people are interested in epigenetics. But again, after fertilization, all these dynamic changes in hydroxymethylcytosine or 5-hydroxymethylcytosine, you just need to take it as, okay, different types of DNA methylation. And the gene that's controlling these changes are called 1011 translocation family, so TET gene. And when we look at these expression profile of the TET family, and we see a significant enrichment in early stage versus late stage. So early stage, it seems like it appears that TET3 is the main gene that governs that process, whereas in the late stage, it seems like it was TET1. So based on this finding, we decide to study or identify, then why do we have TET1? So the easiest way is to remove the function of the TET in the embryos. So we decide to do that, and to disrupt the function of TET1, and I will step back and explain, what's the typical way to remove a function of a gene. At cellular level, typically people use siRNA, small interfering RNA, that can reduce the amount of transcript, or we can apply a knockout approach that we can, meaning we apply genome editing system to modify the genome itself. The effect of siRNA typically fades out, whereas effect of knockout will stay. So we decide to go with a knockout approach. However, this is not a really easy way to, I tried to explain it in a simple way. However, this is really challenging to perform in large animal species because, for example, if you want to harvest knockout embryos in traditional way, you need to make a male knockout and female knockout, and through breeding, you generate these embryos. But again, if it takes over a year or 2 years to generate these pig models and then breed them to get these embryos, it will be very challenging. And if you're working on a PhD, your PhD program is going to be going to have a really long-term plan. So instead, we decide to modify the genome of TET1 directly in embryos. So we introduced a series of CRISPR-Cas9 system, and we performed a direct injection. And we were encouraged by our previous success in generating immunodeficient pigs. We were able to get 100% targeting efficiency. So when we look at the outcome of the knockout, we can see here 5-hydroxymethylcytosine was abolished in TET1 knockout embryo, whereas 5-methylcytosine was not changed. And when we look at DNA methylation level, and this indicates the level of methylation in eukaryotic region, this sign level, it seems like it appeared that a DNA methylation level goes up if TET1 is not there, whereas the centrometric satellite region did not change. And when we look at gene expression profile of pluripotent genes in these embryos, we actually surprisingly saw increase in NANOG and ESRRB, which again, our assumption was that if we block these, we would embryos would lose pluripotency. And so it was an opposite result. And again, more is not always better. So we were trying to understand why. And it seems like when we looked at the expression level of all TET family, TET3 got activated to compensate the action of TET1. So to kind of answer some of these questions, we now introduce an inhibitor that blocks all TET activity and found some similar findings where we do not see 5-hydroxymethylcytosine. Methylcytosine signal was not changed. We saw a similar increase in DNA methylation level in eukaryotic region. And now we finally saw a decrease in most of pluripotent genes after repressing all TET family. So that kind of helped me understand what was going on. So again, the conclusion was inhibition of TET family leads to reduced pluripotency in embryos. So, and basically we were able to tackle that biological question, what does TET1 do? And our hypothesis is that in wild type, TET1 governs all these changes in embryos at later stage. However, if TET1 is not there, TET3 steps up and try to maintain some, but it's not a very accurate way to do it. But if none of them are there, we see a lot of abnormalities in their epigenetic marks. So the genome editing system really allows us to use livestock species for basic research, which again, in basic level, these animals are rarely used because of all these obstacles, and having genome editing technology really facilitate us to use these models. So kind of not summarize, but introduce my ongoing and future directions. Some are funded projects and some I'm seeking funding for future directions. We are interested in safe use of genome editing systems. As I mentioned, these genome editing systems often introduce double strand break. We do not want DNA break to happen, then what do we need to do? Is there any precaution we can use? And we're interested in building better pig models for cell transplantation. And these things, it's really collaboratory work with other groups. They tell me that I'm too ignorant, and I tell them they're too ignorant, but that's how the collaborations work. And I learn a lot of immunology, and they provide me some suggestions on how to improve these models, try to follow and go back and forth, and the feedback is very important. And we try to improve efficiency to produce these pig models. Again, considering the gestation period and everything, every other step needs to be improved for the pig models to be competitive in biomedicine world. And one of the basic line of study that we do is to understand mechanism of oocyte-mediated reprogramming. And other more embryology-oriented studies, we try to explore strategies to produce healthier in vitro derived embryos, and so that we can we understand the biology of pig embryo or livestock embryo development, and hopefully we can apply those to improve animal productivity. Acknowledgement goes to researchers in my lab, I don't have many people anymore, but some postdocs, PhD students, graduate students, undergrad students, and collaborators here at Virginia Tech and Cincinnati Children's Hospital. And the image there that used to be my lab, maybe about a little over a year ago, but with the transition, graduation, and some COVID, I lost most of them. So I'm really starting over. So if you have someone who's interested in this route of research, please contact me. And lastly, again, I mean, all these activities were possible because we were able to obtain and maintain funding from these funding agencies and institutes. So I thank them as well. So I'll stop there. And I keep hearing these beeping sounds, so I don't know how much was delivered, but yeah, thank you.
Well, thank you, Dr. Lee. Yes, we had a bit of a disruption here and there, but for the most part, it came through very well. At least that was my experience, and I hope for the rest of the audience, that was the same. Well, first of all, thank you very much for a fascinating overview of your research on pig models for both biomedical and agricultural applications. And I really appreciate you trying to keep it simple for a diverse audience that we have today. I'm encouraging the participants to post your questions using the Q&A box, but I haven't seen any questions so far. Let me check the box again. I don't see any questions. Let me check the chat room. Sometimes we have questions in the chat room. Well, I don't see any questions there either. Well, Kehoe, this may be your lucky day, but I'm going to ask you at least one question.
Sure.
Well, I do have a couple of questions, but while I ask these questions, again, from the audience, if you have any questions, please use the Q&A box, and I will repeat the questions to Dr. Lee. So, Kehoe, my question is about your biomedical pig models. Do you think having a school of medicine right here on the same campus will be synergistic and help you expand or strengthen your program even further?
Oh, absolutely, absolutely. I mean, ultimately, I don't want to do something that doesn't have an application point. And a lot of people, we're centered around our world, so I know what I'm good at. Often cases, I don't look at any other things, and that's where the collaborations are great. And distance sometimes matters because you just sit down with them, and again, yelling at each other, saying, "Okay, this is the way to work." And they often tell me, "Hey, this is what really works in clinic." Then I sometimes revisit my approach and say, "Hey, how about this?" And that's how we try to generate something that's relevant in clinics.
Okay, great. Well, we are so excited that you have come to CAFNR. So did you say when we were talking before we got started that today is your first day? Did you say that?
No, I started in September, but yeah, not less than 2 months.
Yeah, that's what I thought. You started back in September. So, but I thought you said something along those lines. So that was not what I heard then.
So some of the internet thing, they arrived today. So I think that was
That's what you meant. Okay, okay. Well, so we do have a question here, and that was kind of related to the second question that I was going to ask, but I will ask that again, but let me ask the question that is posted here in the Q&A box. That is, what do we know about the consumer's response to gene-edited livestock?
I think it depends where you ask, which region. It really varies. I know a few companies are trying to get FDA approval. I can share my personal opinion, but it really depends. But then, as a person who's working on these models and focusing on USDA missions, if you look at the trajectory of human population and the productivity that we maintain, we need to do something dramatic to make sure that everybody's fed in a few years.
So on a related topic, when you talk to the public, most of them don't really understand the difference between a GMO and a gene-edited plant or animal.
Right.
So how do you overcome that? Well, in the scientific community, we understand that, but how do you really tell that story to the public?
And I often have these conversations with my colleagues, and we reach to a point that we're just going to have to be facilitators or promoters for these events, and really, not just sit around in our offices or just go to conferences where everybody understands what's going on, but reach out to more general public and say, hey, no, that's not right. This is really different, and why it's different, and more in a simpler term. Don't really, just throw a scientific jargon there and expect everybody to understand. But I think those reach out and extension side is very important for us to be able to utilize these animals in agriculture.
Okay, well, I have one more question posted here in the Q&A box. That is, how long were you able to grow the organoids until they hindered the pig's overall health?
Well, so that's a good question. So the longest transplantation period we had was, I believe it was 10 weeks, and pigs were fine. So our experience is that, again, I mean, these pigs are fine as long as we keep them out of any pathogens. So we either lost pig in a week or we kept them alive. So the last run with some more, I guess, experience, we lost, I think, one after surgery, but the rest until the end of our research period, we did not lose any. So I don't believe there is a really limit to it, unless, which is not the case. So it is really, again, depends on how we raise them. And as long as these organoids are cultured or generated in sterile environment, they should not cause any problem to health of these pigs.
Okay. Well, the last call for any questions for Dr. Lee. Well, I will use the 6 seconds rule. Seeing none. Dr. Lee, thank you again. I know you are in the middle of settling down here in Colombia. So I appreciate it very much taking the time and talking to our faculty, staff, and students today. So I wish you the best. And also would like to thank our audience. I know you are all busy, but thank you very much for taking the time to listen to Dr. Lee. We will continue with our webinar series. We have one more speaker scheduled, and that is Robin Rotman, assistant professor from the School of Natural Resources. She will be speaking to us on November 12th in 3 weeks. And we'll be announcing that seminar and her topic in the very near future. So again, thank you all very much and have a good rest of the afternoon.
Thank you.